I'm Josiah Helmer, the one and only, and this is...
I'm Katryn Gatchell.
We did a research paper. No, we didn't do it. Our research paper that we are going to present to you is on a humanized antibody inhibitor for cathepsin L.
This was written by Xiaojing and Yong.
A little bit of the background about cathepsin L and why it's important. Cathepsin L, also known as CTSL, is a protein or is a cysteine protease that participates in multiple cell functions, a few of which are protein degradation, post-translational modification, cell death, extracellular matrix remodeling, autophagy, and some immune responses as well. Now, apart from these cell roles that it plays in, it also plays a role in disease initiation and propagation. A few of these diseases include liver fibrosis, diabetes, cancer, cardiovascular disease, and a few inflammatory diseases, among other things. The fact that it plays such a big role in these diseases makes it a prime target for drug development, and the more inhibitors can be used to, or inhibitors need to be found to inhibit this enzyme. So I'll just leave it at that, making it an appealing enzyme for drug development and inhibitors. Now, previous inhibitors have been found, but they've all been small molecules that lack specificity, meaning that they also bind to cathepsin S and cathepsin B and bovine trypsin and other proteins that we do not want it to bind to. However, the previous inhibitors have been potent because they bind at the catalytic sites of cathepsin L and these enzymes, but they lack the specificity. Now, monoclonal antibodies are a new and exciting way that we can inhibit cathepsin L and other enzymes because they are very potent and have high specificity. Be further modified to enhance those effects.
However, using monoclonal antibodies is not always super effective. The binding epitopes that are used from antibody libraries and stuff sometimes lead to binding far from the protease catalytic site, which results in no inhibitory activity. Since the antibody binds far away from the catalytic site, the enzyme is still able to bind to its substrate and perform its normal function. To combat that, in this study, they looked at combining the pro-cathepsin L, which is just the propeptide for CTSL, to a synergist antibody. They used a flexible GGGGS linker, which is just an amino acid sequence, to combine the propeptide to the N-terminus of the light chain on the synergist antibody. For this, they created a fusion antibody inhibitor, and through the study, they were able to find that the propeptide extension at the end of the antibody inhibitor blocked the catalytic site on CTSL, and they also discovered that it was very specific and very potent. They looked at or studied the inhibitive capabilities of the synergist LCNT CTSL PP in antibody by using fluorescence-based assays and also by using surface plasmon resonance analysis to determine how potent the antibody inhibitor was and also how specific it was.
To explain a little bit about the creation of this fusion antibody, figure 1 from our paper shows a little graphic of how it was created.
To create this fusion antibody with the propeptide and the synergist antibody, they used the overlap extension polymerase chain reaction method, which is a way to amplify the number of gene fragments and then combine two different genes using the overlap extensions. In this model, they used an amino acid linker that's highly flexible to fuse the light chain N-terminus of the synergist antibody to the cathepsin L propeptide. This fusion gene was digested and then extracted through a DNA gel extraction to retrieve the DNA segment that was created through the overlap extension polymerase chain reaction. Once the DNA segment was extracted, they used the T4 DNA ligase to insert the segment into a plasmid, which is just a circle of DNA which can be inserted into a cell cytosol, and it can replicate itself separate from chromosomal replication, and in doing so produce a large amount of the protein being encoded in the plasmid. So the DNA segment for this antibody propeptide fusion was inserted into a plasmid, which was then inserted into the cell cytosol, allowing the XP293 cells to express this new protein. And in the study, they were able to harvest the CTSL synergis antibody and then purify it through protein G chromatography to further study it.
And figure 3 from our paper shows a few graphs that show the activity and the specificity.
Enzyme inhibition assays were used to determine how effective the fusion antibody inhibitor was, and these enzyme inhibition assays, various enzymes were diluted with assay buffers and mixed with a fluorogenic peptide substrate to determine how effective the fusion antibody was at inhibiting the activity of the enzymes. So first, in the CTSL inhibition assay, they had the fusion antibody, the CTSL enzyme, and a fluorogenic substrate. If the fusion antibody inhibitor is inhibiting the activity of the enzyme, we should see a decreased level of fluorescence as the antibody will be blocking the CTSL enzyme from cleaving the fluorogenic substrate, which we do see in this graph. We see that with an increased concentration of the fusion antibody, there is a decreased level of fluorescence seen, showing that the propeptide synergis fusion antibody is in fact blocking the catalytic activity of the CTSL. In the CTSS inhibition assay, we are looking at if the fusion antibody will block activity of the CTSS enzyme, which is very similar to CTSL. The fusion antibody does have some inhibitory effect on CTSS, but it's about 100-fold lower than its activity against CTSL. In the CTSB and the bovine trypsin inhibition assays, we see little to no inhibitory activity. There's not much decrease in the fluorescence level. So these enzyme inhibition assays show not only that the fusion antibody is effective at inhibiting the activity of the CTSL enzyme, but they also show that the antibody is highly specific to blocking the activity of only CTSL, and it will not block activity of other similar enzymes within the body.
Now, surface plasmon resonance was another method used, and that is shown in figure 4 of our paper. Surface plasmon resonance is a method used to determine the affinity of the enzyme, or to determine the affinity of the inhibitor for the enzyme. Surface plasmon resonance includes a mobile phase and an immobile phase, much like thin layer chromatography or any other chromatography method. The immobile phase consists of a gold plate with our new fusion antibody enzyme attached to it, and then a buffer, the mobile phase containing a buffer, and CTSL was run over top of that gold plate, and whenever the inhibitor binds to the enzyme, that new molecule then reflects light in a different way than either molecule separate from each other. So a laser and a detector was able to be used to determine how many inhibitors were bound to the enzyme at a point. The inhibitor was dose dependent. We see here in the graph that at 180 nanomolar, the response units were roughly 20. However, at threefold lower, at 60 nanomolar, the response units were cut in half. So it being dose dependent is significant for therapeutic use. Now, in the middle of the graph, we have a vertical line, and that shows when a new mobile phase was run over top with a new buffer and different composition, and that phase released some of the inhibitors from the enzymes, and the dissociation constant was able to be found from that. Now, it should be noted that not all of the inhibitors were removed from the enzyme, only a few, and we see that here after the response units went down only a few units and not all the way back down to 0. I mentioned the dissociation constant earlier. That is important because the dissociation constant is higher than the inhibition constant. Now, a lower k value means a greater affinity, and a greater affinity for the inhibitor means that the inhibitor will bind with greater affinity to the enzyme. If the inhibition constant was greater than the dissociation constant, then the inhibitor would not bind as well to the enzyme. In conclusion, in the study, a new inhibitor for CTSL was formed through an antibody and a propeptide of cathepsin. And this new inhibitor proved to be more potent and stable and specific than previous inhibitors had shown. Future studies, however, need to more accurately determine the stability of this new fusion enzyme or this new fusion antibody, and also study it in a more natural environment in an actual cell to study its therapeutic potential.